Welcome to my video about epistasis. Epistasis can be described as the interaction between different genes, and how one gene can mask or modify the expression of the other. What is important about epistasis is that we can actually end up having multiple genotypes express the same phenotype. The one that we will focus on in today's video is the duplicate dominant epistasis. In duplicate dominant epistasis, we have our first phenotype, seen by the red box. If enzyme A is present, seen by the big A allele, then we are able to move on to our second phenotype. But if enzyme A is not present, seen by the two little a alleles, then we are not able to move on and are still at the first phenotype. The same goes for enzyme B. If it is present, we can move on. If it is not present, we are still at the first phenotype. My ceiling light is enzyme A. If it is on, then the enzyme A is present, seen by the big A allele. If the light is off, then that means that the enzyme is not present, seen by the two little a alleles. Enzyme B is my desk light. If it is on, then the enzyme is present. If it is off, then the enzyme is not present. The two phenotypes are this card being visible and this card not being visible. Here are our four genotypes: ceiling light on only, both on, desk light on only, and both off. Here is the updated map for duplicate dominant epistasis. Here are the four genotypes and our observed phenotypes. Here we are crossing two individuals who are heterozygous for both genes. With no epistasis, we would have four expressed phenotypes in a 9:3 to 3:1 ratio. Now let's compare this to duplicate dominant epistasis. In duplicate dominant epistasis, we observe that both lights on, the ceiling light only on, the desk light only on actually led to the same phenotype. So let's label that. To label, let's take a look at our map. If there's a big A present, we go to visible no matter what happens at the B. If there's a big B, we go to visible no matter what happens at the A. If they are both dominant, then we go to visible, and if only they are double recessive, do we stay at not visible. Here, we can combine the first three genotypes as one phenotype and conclude our 15:1 ratio for this dihybrid cross with duplicate dominant epistasis. Let's look at a cross between these two genotypes. The offspring will have these four genotypes, and let's take a look at the map to see what their phenotype will be. Big A, in both of these cases, will automatically take us to the visible, no matter what happens at the B gene. Over here, the little a will not let us go to the visible, but the big B will allow us to go to the visible phenotype. Double recessive means that we do not have either enzyme present, neither light is on, so we are stuck at not visible, leading to our 3:1 visible to not visible ratio for duplicate dominant epistasis. Here is a little shortcut to remember duplicate dominant epistasis.